The advantage of using organoids over traditional 2D cell lines is that organoids are derived directly from the patient's own tissue, and they resemble the genetic makeup and phenotype of the patient, offering a more realistic platform to model the tumor and test a drug response and possibly resistance. Hi, my name is Yasmine Abouleila. I work as a senior scientist and project leader at HUB Organoids. I lead multidisciplinary projects within the oncology space, and my role in PERSIST-SEQ is to lead and coordinate our contribution from HUB side. So initially, we were standardizing and optimizing the processing and how to culture and deal with organoids to have like a standard operating procedures for all the consortium partners to maintain consistency within the experiments across different labs. As the project progressed, we were able to generate drug-tolerant persisters from patient-derived organoids towards standard of care treatment to different cancer types. So organoids are lab-grown 3D cells that are obtained from patients' tissues, where we isolate stem cells, and then these stem cells have the unique ability to kind of self-organize to structures that still capture or mimic the same characteristics that the original tumor has. We use these organoids for modeling diseases, like in cancer, for example. We want to see how organoids respond to treatment, if they respond or resist to treatment. And also, since organoids are derived from patients' own cells, they resemble an accurate model to test treatment and understand how diseases progress and to develop new treatments. We have successfully tested and refined the processes and procedures to culture and process organoids for downstream analysis like single-cell RNA sequencing. We have generated SOPs to align with all the consortium partners on ways of pre-processing and culturing. And then we also made some good progress with generating drug-tolerant persister cells from patient-derived organoid towards standard of care treatment. The model has a lot of complexity. It captures a lot of the patient tissue complexity, heterogeneity, and then in the end, it's more accurate and personalized approach. At HUB in general, we have an extensive biobank of patient-derived organoids. We have patient-derived organoids from 7 solid cancer types, including pancreatic cancer, colorectal cancer, breast, lung, head and neck, etc. For PERSIST-SEQ, the focus of the consortium is to model cancers with a special focus on breast, lung, and colorectal. So far, we have been focusing on colorectal cancer and specifically metastatic colorectal cancer. So all patient material obtained or used in the PERSIST-SEQ project are obtained from the Foundation HUB Organoid Biobank, which collected, processed, stored the data according to the competent authority in all relevant laws, and patients, of course, provide or sign informed consent that their material or samples are being used and stored for future research purposes. And then us at HUB Organoids BV, we use the organoids derived from these material for the agreed upon purposes, and then of course following the competent authority and ICF. So working with other PERSIST-SEQ partners within the PERSIST-SEQ consortium has really enhanced our research at HUB. Working with such diverse group with different expertise, they bring in new perspectives, helped us improve our approaches, our knowledge and expertise in areas like single-cell RNA sequencing and drug resistance has been key for us to think and refine our PDO-based disease modeling to better reflect how a real tumor behave and how they respond or not to treatments. These partnerships, of course, have pushed us to adopt the latest technologies and try out-of-the-box experimental designs and overall enhanced the relevance of our assays. So one of the biggest challenges has been maintaining consistency in how organoids are being cultured and processed and pre-treated for other downstream analysis, since even small changes in these processes can really have an impact on the outcome or the overall result. So to overcome this, we work together with consortium partners and us at HUB Organoids as the organoids experts. We have worked a lot on optimizing and then refining standard operating procedures to guide the partners and then also ensure alignment across different labs. We also worked closely to refine those protocols and share best practices, troubleshooting, and this really helped us to overall achieve much better reproducibility across labs. So we see a big role for patient-derived organoids in helping guiding clinical decisions. So by testing how patients' tumor respond to different treatments in the lab, we can identify the most effective treatment options tailored to that specific individual. And this can support doctors and clinicians to make more informed decisions and choices. Plus, of course, organoids can help us predict how tumors can change with time and then also develop resistance. So we can stay one step ahead and then keep on developing new treatments that can tackle this problem. For me, the motivation comes from wanting to bridge the gap between research and what actually helps patients, especially in something as complex as cancer. At HUB Organoids, I get to work with technologies that let us model patients' tumor more realistically and accurately, which means we are not just generating data, we're actually creating something that can directly inform on treatment decisions for doctors. Being able to test drugs on models that reflect how tumor might actually behave gives us better understanding and a better shot in finding what could work for someone in real clinical setting. Looking ahead, the field of organoid technology is poised for several groundbreaking developments in cancer research. One key area of focus that we will be accelerating thanks to the additional resources that became available for us as part of the life science business of Merck KGaA Darmstadt in Germany is the development of organoids for difficult to establish tumor types such as some breast and prostate cancers. These models will provide deeper insights into tumor heterogeneity, enabling researchers to study subtype specific drug responses, which is important and relevant for PERSIST-SEQ. Another exciting breakthrough is the potential to conduct co-clinical trials using organoids by generating patient-derived organoids in parallel with ongoing clinical trials. This will allow us to predict patient responses to therapies in real time. And this approach will not only enhance patient stratification, but also accelerating the identification of effective treatments, reducing trial and error in clinical decision-making. These developments, combined with our ongoing efforts in expanding our biobank, modeling resistance, and also maybe integrating a little bit of AI-driven biomarker discovery, are set to, we believe, transform how we understand and treat cancer, bringing us closer to truly personalized oncology solutions. We are currently collaborating with our partner, IRB in Barcelona, to investigate if we are able to identify and replicate drug-tolerant persister markers across different platforms, such as patient-derived organoids and patient-derived xenografts. This will help us better understand how tumor persists and eventually develop resistance to treatment, to find better strategies to tackle this resistance, which is the ultimate goal of PERSIST-SEQ.